Okay, next topic is the blood-brain barrier and meninges and ventricular system. So meninges is a name for the connective tissue for the brain. Let's briefly talk about the blood-brain barrier. To compare the most capillaries in the body versus the brain capillaries, there's a stark difference. It's heavily guarded, in other words, by this astrocyte processes. So it forms also, the capillary forms a tight junction and no pores can be found in the capillary, whereas the most capillaries in the body, there's a small pore, so a small particle like polyparticles such as ions can go through in and forth. Of course, the liposoluble substances can travel freely across the capillaries for both cases because the characteristics of the membrane. The astrocytes are the part of the glial cells. I think it will be covered in next week's revision. So it's a supporting cell for the neuron, and its processes are surrounding the capillaries. So what the function of the astrocytes is to transport the nutrients to the neurons and give back the waste to the capillaries. So it's waste removal plus the nutrient supply that this so that's the function of this glial cell called astrocyte. And also the carrier proteins are involved to transport specific ions and specific substances across the capillary. So in other words, the brain is very well protected from the external substances due to these mechanisms of this blood-brain barrier. What other mechanisms are involved in supporting the brain and the spinal cord? It's the connective tissue, which is the meninges. There are three types of connective tissues: dura, arachnoid, and pia mater. So dura mater, arachnoid mater, and pia mater forms the meninges. Mater means mother in Latin. So the most outer mother or supporting structure is dura mater. So durable mother. As the name implies, it's the thickest connective tissue that surrounds the outer part, and it's closely adjacent to the bony structure, which is the skull, the brain. Beneath the dura mater, there's arachnoid mater. So this part is the arachnoid mater. Arachnoid means spider, and arachnoid mater forms a trabecular system. So there's a trabecular system that projects to the pia mater, and that trabecular system looks like a spider web, and so the name comes, the arachnoid mater. And also because of this trabecular system, there's a space between the arachnoid mater and pia mater. So pia mater is the innermost meninges in the cranial meninges, and that's that envelopes the neural structure. And that's the thinnest structure. And you can find the capillaries in the pia mater that forms the blood-brain barrier that we just covered. So the hollow space between the dura and pia mater, this space is particularly called subarachnoid space, and that's where you can find the cerebrospinal fluid. So you can imagine the space is filled up with the fluid. If you go down to the spinal cord, the same meninges covers the spinal cord, so the outermost will be dura, and underneath dura there's arachnoid mater, and between arachnoid and pia mater there's a sub arachnoid space, again filled with cerebrospinal fluid, and that's the pia mater, the innermost. And then there's neural structure. So in other words, the cerebrospinal fluid is enveloping the entire CNS. There's a slight variations between the cranial meninges and spinal meninges. The difference is in the dura mater. In the cranial meninges, dura mater is actually 2 layers, but for the spinal meninges it's just 1 layer. So this is the diagram that shows the 2 layers in the dura mater in the brain. So dura mater, one is periosteal and the other one is meningeal layer. So between periosteal and meningeal layer, there's that runs the venous structure, which we just covered, which is the superior sagittal sinus that collects the superficial venous blood in the skull. So because the dura mater is two layers, between 2 layers you can find the superior sagittal sinus. The other dura mater structure is shown here. Falx cerebri that divides the left and right hemisphere, and tentorium cerebelli, which divides the big brain to the small brain. Okay, lastly we're going to cover the ventricular system, and ventricles are the site that generates the cerebrospinal fluid. So it's a hollow structure, interconnected structure that we can see in the middle aspect of the deeper aspect of the cerebrum, and the forebrain, the diencephalon. These cerebrospinal fluid is produced from the structure in the ventricular system called choroid plexus. So from the internal carotid artery, the branches from that, the capillaries from that branch is filtered through with a specific protein transport system to form a fluid, and it fills up the space ventricular system, also the subarachnoid space that surrounds the brain. If you add up all the volumes of this hollow space, it's about 150 milliliter space. It's a tiny space. But in the choroid plexus per day, we produce about 500 milliliters per day. So if you think about this, if you don't drain the cerebrospinal fluid, it will just accumulate and push all the neural structure outwards, increasing the internal cranial pressure, intracranial pressure, sorry. So you should also think about the circulation and also the drainage of this cerebrospinal fluid. before we discuss the circulation, let's just label those structures. So the biggest ventricle you can find in the forebrain is this C-shaped structure called lateral ventricle. So this is lateral ventricle. It has anterior horn, inferior horn, and posterior horn. So two sets of these lateral ventricles are present. It's connected to the third ventricle that sits between two thalami, and it is connected to fourth ventricle via cerebral aqueduct. So this narrow passage is the cerebral aqueduct. And from the fourth ventricle, so this is the fourth ventricle near the cerebellum, it drains out to the subarachnoid space. In order to help you visualize this structure, I actually used this diagram in my undergrad degree for my undergrad students. So you can imagine like two sharks. The two sharks, you can draw two sharks here, ferocious sharks, trying to eat a chicken. So chicken, you can draw the chicken so about to be eaten, and this poor scared chicken has its neck down, and the body, chicken body, is forming its fourth ventricle, and chicken feet is the aperture towards the subarachnoid space. So you can just place this. Yep, that's poor chicken. That chicken head now becomes the third ventricle. Chicken neck becomes the cerebral aqueduct. And chicken body becomes the fourth ventricle. And it goes to the subarachnoid space. And the sharks are the lateral ventricle. So let's look at the circulation. So the general circulation direction is shown in this diagram. So each ventricle has the choroid plexus that produces the cerebrospinal fluid. But let's start from the fourth ventricle. So the fourth ventricle is connected through the third ventricle via this hole called interventricular foramina. And it circulates to the third and through the cerebral aqueduct, goes to the fourth. So this is the fourth ventricle here, and goes out to the subarachnoid space. And it's enveloping around the entire brain and the spinal cord. And it literally gives the buoyancy to the brain structure. It's so powerful that if you think about this, the average weight of the brain is about 1.5 kilo, but due to this buoyancy, it's reduced to 50 gram the weight. And this is critical because if you don't have this buoyancy, all the neural structure that sits in the bottom will actually be crushed by the weight of the structure above it, and it will cause the ischemic damage to the neural structure. So by the help of this buoyancy, the buoyancy force, it's reducing the weight so that the structure in the bottom is not necrosized. So that's another important role for this cerebrospinal fluid. Other roles could be waste removal, and also providing the immune circulation of the immune cells throughout the brain structure and so forth. The last part would be how to drain this cerebrospinal fluid out from the brain and spinal cord. This is through the arachnoid granulation. So arachnoid granulation is connected to the superior sagittal sinus. So this is the superior sagittal sinus that is the part of the venous structure in the brain, and the arachnoid mater forms a granulation, and that infiltrates into this space, the superior sagittal sinus, and because the subarachnoid space is filled with the cerebrospinal fluid and the pressure builds up high, this is high pressure versus low pressure in the superior sagittal sinus, these granulations act like a pressure valve. So high to low pressure, the fluid automatically goes into the superior sagittal sinus and drain into the venous system, and that venous blood will eventually joins to the systematic blood circulation. And you can also appreciate this superior sagittal sinus is formed between the two layers of the dura mater, periosteum and meningeal dura mater layers. Okay, so that's it for this mini lecture. So blood circulation and meninges and ventricular system. If you have any questions about the lecture, you can post it in the Leo or email me separately so that I can actually answer it in the Leo when I combine all the answers to your questions. Okay, thank you, and I'll see you next week.